cruciferous is basically the plant family crucifery. Okay, I don't know if you know about it, or we call sometimes the family Brassicaceae. Okay, but commonly known as, and I give you the common name for it, known as mustard family, because one of the species from it we make the mustard. Okay, called the mustard family. Okay. Anybody have an idea what are the crop in the mustard family?
Like cauliflower and like Brussels sprouts and stuff.
Cauliflower, cabbage, all that. Because this one basically, when you eat it, has a special kind of what you call biting your tongue. Okay, which is basically due to this compound known as glucosinolate. Okay, which is a secondary compound. And of course, one of the family, which is a Brassica, one of them is Brassica alba, and the others, in which basically they can collect what you call the mustard. So simply going to mention to you here just one line to put it this one. As I mentioned, that this species causes the disease of the crucifer, okay, which is simply, it is of course an obligate parasite like on horseradish and cabbages. Okay, like horseradish. And cabbages. But in general, okay. Just give me a second here, because this is what happened here. Usually when infect the plant, I'm not giving you the life cycle, but at least to show it to you, okay? When infect the plant, okay, it grows under the epidermis. This is shows to you here. This is a leaf. This is the epidermis, okay? And it grows under the epidermis. And this is what you call the tissue under the epidermis. And then when it produces a lot of conidia, and then of course, it will push the epidermis and comes out on the surface of the plant. It looks like a white powder. And that's what we call white rust, okay? So you follow what I'm saying here. So basically that's how it grows, like this one here. Shows to you here, this is a color picture, okay, of Albugo candida. That's how it grows, okay. So what happened here, when you have the leaf, and then what you call it, because it grows in the tissue, and then it will push the epidermis like this, and in the end, of course, it comes out like a white powder. I think you have a picture of this, okay, yeah. In the end, it comes like this one here. Okay. Do you see this white picture here? Okay. And that's basically what it grows under the epidermis and pushes it. So this basically known as white rust. Okay. That's how it grows. It looks on the surface of the leaf. And basically, of course, here are all, because these, all the pathogen, all the plant pathogen, when they grow, okay, in the end, they produce their spores, whether in case, mostly asexual, they produce above the epidermis, so that basically it can be transported to others. So always they will do that, okay? Though I mentioned two different species, but of course, each one of these, as a matter of fact, they have different kind of, I'm going to show it to you here, okay? Okay. Yeah. So basically, some of them they produce like this one here, of course, this, for example, how it looks like the conidia or the spores of plasmapora. This is how it looks like, for example, of the polyspora, how it looks like. So they have different shape, and that's why we can identify them. Okay. So this basically just I want you to know this one here. They give you this example of what you call Saporigina, Plasmapora, Pernispora, Pythium, Phytophthora infestana, and of course later on I give you this example here, okay, of Syncytium antibioticum and Albugo candida. Okay, so you should know the name and what disease will cause. Okay. Is that clear now? Okay. So now, basically, as a matter of fact, I'm going to move to, I finished already by this. I will end up the section of the oomycete or the Mastigomycota, and we're going to move now to a new group of fungi. Okay. Going to move to the phylum Zygomycota. Okay. Now, all these fungi, as a matter of fact, once you move to the Zygomycota, or we call them the class Zygomycetes, okay. Now, all these fungi, none of them will have what you call forming like a flagella or zoospore. They produce all either a spore in different form, okay? And also one thing, I think I sent you the title of your essay. Did you receive it through DTL?
Yes.
Thank you. Yeah. If you have any question, if you want to just don't hesitate to send me an email if anybody wants more what you call clarification. Okay. Now, these are basically an important kind of fungi. Okay. In general, we're going to take you about their general here. Now, these fungi, I'm just to give you, for example, their characteristic here, one of their characteristic, okay? That these group of fungi, when they do their sexual production, of course, mostly they produce what you call a structure of called zygospore, a thick structure exfol. So all these group of this fungi, they produce this zygospore, and that's why we put them there under the same vertical group as zygomycetes. Okay. Other characteristic also we're going to mention to you later. I will put down here. During their asexual production, they produce a structure. They produce their spore in a form of sporangia. So you have sporangia, and inside it are the spore, and of course, so basically in their asexual production, mostly produce the sporangia, and during their sexual production, they produce the zygospore. Now, also the mycelium or the hyphae of these plants, as a matter of fact, it is multinucleate or cenocytic, and they produce that means there is no septa, only one septa at the top here when before just the sporangia were developed. So basically, first of all, I'm going to give you a little bit of outline of this, okay, of this example. Some species of this group could be important, and some, of course, could also important in sense causing pathogen or so on. So we're going to have different groups. Now, in general, I'm going to mention that most zygomycetes okay, live, okay, on decaying. They live on decaying plants and animal matters. Okay, and the soil. Okay. Some of them, others, okay, are parasites on plants. Okay. Some also they live in symbiosis because some of the species they do the mycorrhizae, so that's what I mentioned here. That okay. Others mean when I mention other means other species. Okay, form called symbiotic association. For symbiotic association. Now we call endomycorrhizae. Mostly is endomycorrhizae. As I mentioned to you before, that this symbiotic association between the fungus and the root we call mycorrhizae. And it's very important because almost like 90% of vascular plant, they need mycorrhizae, some sort, basically, to grow more better. Okay, but as I mentioned, some of these fungi, basically, it grows only outside of the root. In that case, we call ectomycorrhizae. And some species, they go inside, especially in the cortex. We call them endomycorrhizae. Of course, I'm going to talk about it later on after I finish all the fungi and to explain what species grow as endo, which species are ecto, and what is their importance. Okay, so some of the species form this endomycorrhizae, and others, of course, there are also some zygomycete could be pathogenic. Others, of course, cause infection. In a human and domesticated animal. Okay. Now the other characters basically to mention, they are so far about okay, about 1,060 species of zygomycetes are described. Described or identified. Okay. Now, it's important to mention no flagellated. That means, okay, no flagellated. Okay. Cells are found okay, in any of these fungi. So basically, there is no flagellarium in any of these fungi. Okay, the other character mentioned to you here. Okay. This characteristic, most of zygomycetes, hyphae, are multinucleate. That means there is no septa. That means, in other words, senocytic. Okay, However, there are a few species, it could be what you call unicellular or a yeast form. However, I'm going to mention, okay, some of them are in unicellular form. In that case, if it's a unicellular, we call it, of course, like yeast-like. Call them yeast-like forms. Okay. Now, the other characteristic important to understand of this group here, now, as I mentioned to you, that asexual protection, okay, by means of haploid spores, okay, of haploid spores produced in sporangia. Of course, always remember that sporangia is a plural. When you talk about one, in this case known as sporangium, singular is sporangium. Okay, as this one here, as I showed you here. Okay, that basically is for. And of course, I don't know if you can see this one here. Of course, okay, this is the sporangium here, and of course, it has a very delicate. I want to talk about that. It's a very delicate membrane here. Once the spore are mature, it's going to the membrane is going to tear out, and of course, then all the spore are going to be what you call widespread by the air. Okay. Now, this species, of course, one of the major species. This one is rhizopus, which is very common all over any area you find it there. Okay, and causes what's known as some of the species cause what you call the black mold of the bread. I'm going to show it to you later on here. Okay, so but at the moment, that is basically the asexual is by the sporangia or sporangium. Okay, born on a hyphae. Okay, so simply as I mentioned to you, okay, that these species always you have this terminology here that if this is the hyphae, okay, and this is when you have the erect hyphae like this, okay, and here is the sporangium, okay. Now this is the going to find this section. So inside, as a matter of fact, okay, these are the spores. This is the sporangium. Okay. Now, this part here, going to see, I'm going to talk about, which is known as the sterile part of the sporangium. You know what you call it, maybe from it's known as the coliomella. Okay. And normally, the hyphae which carrying the sporangium, it is known as the sporangiophore. Okay. And of course, usually with more, so you're going to find also these ones, sometimes they have rhizoids, some of the species here, okay? Which is sometimes, I'm sorry, if you have really the media, one can show it to you, but at the moment, of course, we cannot. Okay, though even with the under the microscope, but you need very careful examination, you can see the rhizoid digging inside the agar. Okay, so anyway, as I mentioned to you, let's continue the sentence. I will just so that you understand that. So asexual production by means of the haploid spores produced in a specialized sporangia, okay, which I mentioned sporangium, okay, born on a hyphae known as, sorry about that, on a hyphae, and this hyphae known as sporangiophore. Okay, sporangiophore. And basically, this is almost universal in zygomycete. As I mentioned to me, is almost universal, means occur in all the group of the zygomycete, and the group of zygomycetes. Make sure that you can see it here. Okay. Now, as I mentioned to you, basically, that the whole group of zygomycete. Now, just put down here that this phylum Zygomycota, okay, is named, okay, for its main or chief, okay, characteristic, okay, which is the formation of sexually produced resting spore, okay, resting spores, which is known as it called, mentioned you called the zygospore. So that's why basically we call it zygomycete, because during their sexual production, they produce this, what they call the zygospores. Okay? Okay? What other thing here to mention to you? What other point going to mention here? Okay. One of the best known example of this group here, okay, one of best example, we know is Rhizopus, okay, Stolonifer. Okay, best example of this occurrence. This is a typical example, which look like this one here. Okay, normally when it grows in a petri dish, like this one here, okay, do you see this one? Okay, usually it gives a very black, of course, all the time, remember, do you see this one here? This is the hyphae, of course, the hyphae itself looks cottony white. All the color you see of the fungi because of the spores. Okay, could be blue, could be yellow sometimes, or red. And that's where when the spores are produced, or the sporangia, basically it is black in color, and that's what gives you what you call this black color here. Okay, which is commonly we know the Rhizopus stolonifer, also known as black mold of bread. Okay, give me a second here. Okay. The other thing I want to mention to you here, okay. This what you call going to start with you here. The class Zygomycete includes 7 orders. Includes 7 orders. Okay. But we may be able to discuss 2 or something like that here. Okay. I think I have a somewhere here. Just a second. Because all the time, I prefer to show you the transparency on scientific here. All right. Now, I'm going to discuss mainly 2 orders of this one here. One, the order going to, basically one of the order going to focus is the order, what you call Mucorales. And the order going to focus on here, okay. Okay. As I mentioned, there were 7 orders, but I'm going to focus more on the order Mycology. Okay. Now, in this phylum, as a matter of fact, the whole phylum, the phylum, what you call Zygomycota, we have 2 classes. One class, as I mentioned to you, known as Zygomycete, which is more important. Okay, and in Zygomycete, these are the classes. Let me, okay, put you here. Zygomycete class, we have the order. Orders, you have Mucorales. We have the order Intibophteralis. This is A. And also we have the order Kibzelialis. And we have the order Glomialis. Okay. And the other class, this is one class important in the phylum Zygomycota. The other class known as Trichomycetes. Trichomycetes, sorry, put it here, class. Yeah, class. We have here Trichomycetes. Trichomycetes is it contains a very large group of fungi, but most of them basically we call them, because economically not important. Economically not important, what is the term? They don't cause any kind of disease. Most they can find in insects, okay? All of them, okay? And some of them are so specific, we're going to touch on it at the end, okay? And specific in a sense, you find some species choose between insect only the male or only the female, but they can live just in a harmony with causing nothing, okay? So these are basically going to talk about here. So this is basically as a general about this group here. Now I'm going to start with the first group as an example, okay? We're going to start basically with the order, which is important, order Mucorales. Okay. As I said, the most example we're going to use in this case from the order is Rhizopus stolonifer. Time passes very quickly here. I have a lot of material. Are you following it okay here? Okay.
Yeah.
Yeah, and which is known as I told you, this species known as the black mold of bread, commonly known as black molds of bread. I'm going to use it as an example, basically to describe it as an example of the group of the, okay? Though there's an example in euchre, you, of course, you cannot see the percentage, maybe the, I don't know if they show it to you in the lab, the percentage of, did you see the percentage of rhizopus in the lab or not yet?
Yes, yes.
Yeah. A euchre differ only basically it looks more yellowish and a little bit the stone. I'm going to give you the stone a little bit different, but more or less exactly the same thing here. Now, just to talk about this one here, okay? These fungi, of course, rhizopus could be found, can grow on over. Now, uh, as you know, because the fungus basically lives on the way it absorbs its food through enzymes. So if that fungus can live in many places, that means it have enzyme can consume many kind of substrate, and that's why you find, for example, rhizopus is one of these are all over. That if I take a petri dish now and leave it in the air just like that for about half an hour and then incubate it, definitely is going to show a rhizopus there. Okay, the same thing with penicillium and asparagus, but they are from different groups. These are three to mention this one. Okay. To mention the other good to mention the mycelium. Okay, the morphological structure of this organism. It's mycelium. Okay, its mycelium is non-septate. Okay, which was it? And may be present okay on all sort of organic matter. All sort of organic matters. Okay, like for example, like dung. Okay, fruit, and also on a fleshy fungi. Okay. Uh, and as I mentioned, and it's spore, okay, it's spores, okay, almost and always, to mention to you here, present in the atmosphere. Okay. Now, the mycelium, as I told you basically in the beginning, is cottony white. But once the spore becoming give that a black appearance. So the mycelium is cottony white. It's cottony white, okay. But okay, presents, we call it sooty appearance, become black appearance, but present sooty appearance okay, at the time of sporulation. At the time when they do the spore of sporulation. Of sporulation means formation of the spore. Okay. Now, as rhizobacus can show us a little bit of some differentiation here. We're going to that usually, if it grows in the media, it grows like this. It will form a stolon, a hyphae like this, and then in the end, this area is going to use rhizoids, and then of course, opposite to the rhizoid grows what you call these sporangia, which you call the sporangiophore. Yeah, sporangiophore and the sporangia, and here you have the spore. So basically, this is it shows to you this kind of a growth here we call stolon. Can you see that? Okay. So basically, that's what I'm going to cover at the moment here. Okay. Now, to mention this point here, that the mycelium, or this mention here, okay. The mycelium exhibit or shows, okay, considerable or just certain is more, okay, certain types of differentiation as some hyphae, okay, it creeps over, okay, over the substance or the substrate, over the substrate, okay, forming what you call stolon-like branches, forming stolon-like branches. The one I showed to you here. Okay. And also, okay, form, and some branches and also form, okay, absorptive rhizoid. Absorptive rhizoid. That means rhizoid can absorb material. Absorptive rhizoids. Sorry. All right. That clear? Now I'm going to talk about basically the life cycle. Of course, the life cycle, we're going to discuss asexual life cycle or reproduction and sexual reproduction. Now, very simply in the asexual reproduction, just give me a second here. After a period of growth or vegetative growth, then certain hyphae develop what you call erect branches, which is this one here, which is almost opposite to the rhizoid. And then, of course, in the end, we'll be going to develop what you call a sporangium. And inside, of course, the cytoplasm is going to divide and form the what you call the spores. And when it matures, of course, the sporangium is surrounded by a very delicate membrane that is going to tore, and then once the spore are spread, it's going to grow to a new individual. So basically, that is presented what you call as the asexual production. Talk about this one here, okay? That after a short period of somatic growth, after a short period of somatic growth, okay, group or groups, as a matter of fact, groups, okay, of unbranched, okay, of unbranched, elongate, hyphae, arise, okay, opposite of rhizoid, as I mentioned to you. Opposite of rhizoids. Okay. Of course, this hyphae or this erect hyphae, okay, is the sporangiophore, okay, and each bearing okay, a single sporangia. Bearing a single sporangia. Okay, now upon sporangium maturation, okay, a large number this is a large number of minute spores okay, minute spores that this one each one contain what we call many nuclei. Okay, each contains several nuclei. Okay, now the outer wall the outer the outer sporangial wall, as I mentioned, is extremely is extremely delicate. Okay, it's extremely delicate. Okay. And readily torn and readily, what do you call, torn and when this occur, as a matter of fact, the exposed spore the exposed spores, okay, are quickly carried by air current. Are quickly, okay, carried by the air current or by air currents. Okay.
Sir, could you move your page up a little? We can't see what's at the bottom.
Do you see it now?
Thank you. Yep, we can see it.
Okay. By air current. Now, of course, as I mentioned to you, that basically because inside the sporangia there is what you call the columella. So basically, when the air is take all the spores, the only left inside is the columella. Okay, so basically, as I showed you before, I guess, yeah, because that is basically when you see in this picture here, that if you focus on it, yeah, that this part here is the columella. So at the end, when all the spores gone, only the columella will stay. So basically, that's what I want to mention to you here. Air current, and as I mentioned to you here, now the naked columella, define with the naked, without, okay, the naked columella, okay, remains. So the columella basically is the what you call is the central, we call central sterile part of the sporangia. So the naked columella remains. So this is basically with regard to what you call to the asexual reproduction. Now I'm going to move to the sexual reproduction. I think I have only few minutes here. As a matter of fact, this is but I'm going to talk about it now, and mostly I think I will what you call give you the note maybe next. It's better to explain to you now. This table here with really many things. Yeah. Now, just give me a second because you pay attention to understand it. Okay. I don't know how much they describe you now. This organism, as a matter of fact, okay, it is a heterophallus. Heterophallus, that means really we don't have male gamete and female gamete. Usually, sometimes become two strains. When they come together, then in that case, we call one strain as plus and the other strain as minus. So now, and usually what happened here, it seems there is a chemical compound called progamon, which basically stimulate that process. Now, when this is, it shows to you here, if you can see to me here, okay, do you see this one? This is shows a hyphae of plus and a hyphae of minus coming together. Then gradually, near the proximity of each, a protrusion occurs from the hyphae. Did you follow this one? Now, this protrusion in the beginning, we call a progametangia. First of all, occur progametangia. Now, when the progametangia, they grow together, once they touch, each one would touch the surface, then what happened here, when they touch the surface, then in the end, what you call a septa will occur. So if I draw this one like this one here, give me a second. If this is one hyphae, like this, and this is the other hyphae, like this will become touch with each other. Now, when they touch, and then later on a septa will develop here, and septa will develop there. Now, when the septa develop, then this is known as gametes now. Okay, and this part we call it suspensor. You follow that? Okay, did you follow this one? Okay, I think I showed to you here. Okay, so basically that's what happened here. Now when they touch, then it becomes what you call that septa, and now we call gametes. Of course, gametangia is plural here, and this is the suspensor. Now, once these two surfaces touch, then in the end, what happened? That surface is going to dissolve, and of course, once it dissolves, it becomes like one compartment. And once it becomes one compartment, and as I mentioned, that in rhizopus or oligomesid, it is multinucleate. So you have large number of nuclei swimming in this what you call area. But in the end, each two nuclei from plus and minus, they unite, and each one will form a zygote and become a zygospore. Okay. Now, all the nuclei which are not going to unite, they're going to disintegrate. And that's why later on, the whole structure will develop around it, a thick wall, and contain what you call known as a zygospore. Now, when the zygospore occur, okay, now the organism here is haploid, and when the zygospore occur, then of course meiosis will take its place, and then of course will germinate into what you call a sporangium, and of course the spore is going to be in this case what you call haploid spores, and when are basically blown by the wind is going to grow again into the what you call a new organism. So did you follow this? So basically what you happen in the sexual production, very simply before it will stop, we have progamitangia, it becoming gametangia, then they will unite, okay, and then develop zygospore, and then of course the zygospore okay, with meiosis,